Neurotransmitters serve as the primary messengers in neural communication, transmitting signals between neurons by binding to specific receptors on target cells. This binding event initiates a cascade of intracellular processes, known as signal transduction pathways, which ultimately modulate neuronal activity, synaptic strength, and gene expression. Through these mechanisms, neurotransmitters exert profound effects on brain function and behavior, orchestrating everything from immediate responses to long-term adaptations. A major class of neurotransmitter action involves ionotropic receptor pathways. Here, neurotransmitters like glutamate and GABA interact with ligand-gated ion channels, such as AMPA and NMDA receptors for glutamate or GABA-A receptors for GABA. Upon neurotransmitter binding, these channels rapidly open, allowing selective ions to flow across the postsynaptic membrane. The resulting changes in membrane potential can lead to depolarization, producing excitatory postsynaptic potentials, EPSPs, or hyperpolarization, resulting in inhibitory postsynaptic potentials, IPSPs. This form of fast synaptic transmission is essential for instantaneous neuronal responses and underlies critical functions such as sensory processing and motor coordination. In contrast, metabotropic receptor pathways involve a more intricate sequence of events. Many neurotransmitters, including dopamine, serotonin, norepinephrine, and acetylcholine at muscarinic receptors, act through G-protein-coupled receptors, GPCRs. Activation of these receptors triggers diverse intracellular signaling cascades. For example, the KMP-PKA pathway is initiated when certain GPCRs stimulate adenylyl cyclase, raising cyclic AMP levels and activating protein kinase A, which then phosphorylates target proteins to influence ion channel function, gene transcription, and synaptic plasticity. Alternatively, activation of the phospholipase C-PKC pathway leads to the production of IP3 and DAG, mobilizing calcium from intracellular stores and activating protein kinase C, thereby regulating neurotransmitter release and long-term neuronal excitability. Some GPCRs, such as D2 dopamine or A2 adrenergic receptors, inhibit adenylyl cyclase via GIO proteins, decreasing CAMP and PKA activity, and often leading to inhibitory effects by direct modulation of ion channels. Beyond these, certain neurotrophic factors like brain-derived neurotrophic factor, BDNF, act through receptor tyrosine kinases, such as TARK receptors. Ligand binding prompts receptor dimerization and autophosphorylation, activating downstream pathways including MAPK, ARC, PI3K, AKT, and PLC. These signaling cascades are pivotal for regulating neuronal survival, differentiation, axonal growth, and synaptic plasticity, thus playing a central role in learning, memory formation, and neuroprotection. Adding another layer of complexity, some neurotransmitters and neuromodulators function as retrograde messengers, with endocannabinoids and nitric oxide being notable examples. These molecules are synthesized and released from postsynaptic neurons, traveling backward across the synapse to influence presynaptic terminals. By modulating presynaptic calcium influx, they fine-tune neurotransmitter release, thereby adjusting synaptic strength and contributing to the dynamic regulation of neural circuits. Altogether, neurotransmitter-induced signal transduction pathways encompass both rapid electrical changes and slower biochemical cascades. Through these diverse mechanisms, neurotransmitters not only determine immediate patterns of neuronal excitability and synaptic transmission, but also drive enduring changes in gene expression and neuronal connectivity. In this way, they orchestrate the complex interplay of neural networks that underlies cognition, emotion, and behavior.